And this is what you get. So you get A plus, A negative, O plus or O positive, and O negative. So those are your gamete combinations. Okay. So I'm gonna leave that. I'm gonna leave that there with Nelson. I'm gonna go to your key. And we're gonna do a couple questions from your key. Okay, let's look at page, this is in your key, page 98. And the question says, in garden peas, the allele for tall plant height is dominant over the allele for short plant height. And the allele for axial flower position is dominant over the allele for terminal flower position. Okay, so this says a plant heterozygous for both traits, so this right here, okay, is crossed with a test cross. What percentage of the offspring produced would be expected to display at least one of the dominant traits? So again, you have little t, little a, and then you make all of your gametes here. And right, so you see, when you do that, 75% of all these offspring would be dominant for at least one trait, and there would be one test cross produced from that. Okay. Alternate forms of genes are known as alleles. We know that. Here's another one. Question 99. Sorry, page 99. Question number 27. This says, in the hypothetical pedigree below, shaded individuals have sickle cell anemia and are homozygous for the defective HBS allele. So they're HBS. Carriers of the HBS allele are not identified in the pedigree. So this says individual 3-1 has blood type A. What could his genotype be? So 3-1 is affected. Sorry, 3-1 is not affected. Carriers are not identified. So this is not going to show as a carrier. This is going to show as unaffected. So 3-1 has blood type A. Okay, so what could he be? Cannot be B because this would be sickle cell. This would be AB blood type, so that's not A blood type, and this is AB blood type. So the only option is A blood type, carrier for sickle cell anemia. Okay. So that's a bit tricky because it only shows individuals that are HBS, but you should know that blood type is codominant. Okay, and that's kind of your key thing. And carriers are not shown here, so they're shown as unaffected. So a little bit of reading comp, highlight a few of those things. All right, page 100 and 101. Very good questions that I'm going to go over here. Number 33, 34, numerical response 6 and 7 is what I'm going to discuss. So this is polygenic traits. These are alleles that control other alleles. This is an epigenetic question. Okay. So this says 2 genes control the expression of kernel corn and Mexican black corn. Black pigment gene B and dotted pigment gene D. Okay, so I'm going to write that down. Okay, this is page 100 in your key. This is number 33, 34, and NR 6 and 7. Okay, so in black corn, B stands for black, and you could have a dotted phenotype as well, and that's big D. Okay. Gene B influences the expression of gene D. So here's the deal. The dotted phenotype appears only when gene B is in the homozygous recessive state. So if we have little b, big D, then dotted will show up. Okay, if there is one big B, okay, no dotted. So B controls the expression of D. Okay. So a colorless variation occurs when both genes are homozygous recessive. So in little b, little d, right? So this guy would be black. So anytime there's a big B, it's black regardless. But this one's colorless. Okay, so the question asks, after pure breeding, black pigmented plants were crossed with colorless plants. All of the offspring were black pigmented. So you have true breeding, black pigmented plants. So the question is asking, which genotypes are true breeding? So this 33 is pretty easy. This is the only gene combination that are true breeding, right? You have heterozygosity and everything else. Right? Big B, little b here. There's big B, little b here. So there's heterozygosity with B, C, and D. Right? Both alleles are homozygous in A. Okay, so this says plants of the F1 generation are suspected of being heterozygous for both genes. A test cross of colorless plants with a heterozygote plant should produce a phenotypic ratio in the offspring of what? Okay, so in the F1, you have big B, big D, little d, and they are crossed with a test cross. So they're crossed with colorless plants. So we're going to put these together and see what the phenotypic ratios are going to be. So you have BD, right? And then I'm going to get big B, big D, big B, little b, big D, and little b, little d. Okay? So what do we make? So that's black. That's black. That will be dotted because B is in the homozygous recessive condition, and that will be colorless. So you should have 2 to 1 to 1 to 1. So your answer should be C. Okay? So the question is asking, what is the probability of dotted offspring being produced from the test cross described in the previous question? So about 25% should be dotted. And the other question 7 here says if the total number of offspring produced in the test cross was 1,024 plants, how many would you expect to be black pigmented? So 50% of them should be black. So half of 1,024. Okay. Okay, I'm going to do 2 more questions, and then I'm going to leave the rest for practice for you, and then I have 1 more lesson that I'm going to post, and that'll be it for Mendel. This is a very, very good question. So this is page 103 in your key. This is Punnett square for a dihybrid cross. So the Punnett square is a 16 square Punnett square, but it's been inverted. Okay, so this is another epigenetic question. So this says coat color in mice is controlled by the interaction of 2 genes. 3 phenotypes result: black, brown, and white. So So you can see big B is black, little b, little c, big C is brown, and anything that has recessive little c's are white, which is color absent. So here's what this looks like. So if I have big B, C, then we have black. Okay? If we have little b, big C, then that's brown. But if we have anything that's homozygous recessive, that is colorless. Okay? So that's what I'm deciphering from that. Okay? So it says 38, in the dihybrid cross between the two black mice, the C allele codes for, so if there's one big C, color is present. Okay? So there's your key questions. I'm going to give you a question from a practice diploma as well, and then that'll be it for Mendel. Just need to find it first. Okay. This is the question I want to address. Actually, I'm going to give you two more questions from diploma exemplars. Okay, so this question says the inheritance of color in domestic pigeons involves several genes. The dominance hierarchy of three color alleles is ash red, blue black, and brown. These alleles are carried on the Z sex chromosome. A male pigeon has two Z sex chromosomes, and a female pigeon has one Z sex chromosome and one W sex chromosome. So there's a couple things that are weird here. There's a dominance hierarchy, which you saw with the question about co-color in rabbits that I gave you in the Nelson textbook. But birds and reptiles have weird chromosomes. So we have heterochromy in males, meaning that they have different chromosomes, right? So we have X and Y with sex chromosomes. But in birds and reptiles, females have heterochromy. So this is different from what you're used to. All right? So first and foremost, you have ash red. Right? So I'm going to make this CR. And then blue, black. So I'm going to assign this CB. Okay. And then brown. And I'm going to assign this CBR. So, right, so the ash red is dominant over the other two alleles. But you also have ZZ is a male. And ZW is a female. So that is different from what you're used to. So this is why this is challenging. So you have a sex-linked trait and then an autosomal trait. And then your sex chromosomes are different from what you're used to with this question. Okay? So the question asks, which of the following rows indicates the expected appearance of male and female offspring of the mating of a blue-black female pigeon and a brown male pigeon? And my apologies, these exist on the sex chromosomes. Okay, and they are carried on the Z. So this would be ZR, ZB, and that would be ZBR. Okay, so what we have is a blue-black female pigeon. So this would be ZB2. W, that's female, and a brown male pigeon. This would be ZBR, ZBR. All right. So 27 is asking what does the offspring look like? So let's make our Punnett square. So you have those are blue-black males. And then you're going to have ZBRW and then ZBRW. And those are brown females. Okay. So your answer is B, right? So male offspring are blue-black because the B allele is dominant to brown. And the female offspring are all brown. Okay. All right, so here's another question related to this. This says color pattern in domestic pigeons is determined by autosomal alleles. The dominant pattern is barred with 2 black bars across the wings and a black bar near the end of the tail. Barless, the pattern with no black bars, results from the inheritance of 2 alleles. All right, so if I look at this, big B would be barred, and little b would be barless. Okay. So if a breeder wanted to determine the genotype of an ash red barred male pigeon, the breeder should mate it with a female pigeon that is what? So the male pigeon is barred, and then he is red. We don't know which one that is. So he needs to cross the male pigeon with a female test cross. And in this case, it's the most recessive trait. And it's going to be the barless female, right? So the female will be little b, so barless, but also brown. So she needs to be brown and barless. Okay? So your answer to that question would be C. All right, so I'm going to end this lesson on dihybrid crosses here. I'm going to give you one more lecture today looking at crossing over and genetic recombination, and then we'll end Mendel there. Okay.